Hey guys, so I'm going to do a quick video tutorial to show you how to do 1.4.5 shifting populations. This can be found in lesson 4, part 5 of module 1 in ecology. So if you go to your modules and you scroll all the way down to lesson 4 in module 1 ecology, you guys can find 1.4.5 shifting populations. So we talked about how different populations can affect others when their growth goes up or when they go down. We learned about limiting factors for their survival, things that prohibit them from growing too large and taking too many resources. And then we talked about the different kinds of growth that we could see, whether J-shaped exponential growth or logistic growth. And we talked about the factors that connect to that growth, so making the growth rate go up or down within the population. Now we're going to do the Food Web Lab. In order to do the Food Web Lab, you need two things. You're first going to click on the Food Web Lab itself, and that will bring you to the actual sheet that we're going to be using for this. Okay. The second thing that you need is the Gizmo, Coral Reefs 2. So in order to get to the Gizmo, you guys will need to sign in through Clever. If you don't remember how to do that, please see previous videos or the previous assignments with Gizmo to see a quick tutorial on how to get there. Basically, you go through the Work High School home page, you go to the Students tab, you click on Clever Canvas and More, and then you go ahead and sign into your Clever. You should see the W Garners page in there. You click on that, you go in there, you get to Gizmo, you click on the Gizmo for Explore Learning, you find My Classroom, and you find Coral Reefs 2 Biotic Factors. So this is the Gizmo we are going to be using. Now, Coral Reefs 2 is a really cool gizmo to look at how different biotic populations affect other biotic populations, and even how some abiotic things can affect them. In order to learn more about each organism, we can click on it, and it'll tell us about the organism, what eats it, what it eats, and then its current population. And we can do that for all the different kinds of fish and organisms that live in this environment. Okay, we can also change various different settings, right? So it's probably going to start you on ocean conditions, and these are the typical ocean condition settings that you would start with. And then, of course, you can change the acidity of the ocean, the temperature, or the storm severity. You can also change other things like land use, right? Things that humans do that could affect the ocean. Also, the amount of fishing that is being done, the amount of disease, right? How disease affects these different populations, and of course, invasive species, what would happen if we added these in to the rest of the population. Okay, so you're going to need this particular gizmo in order to do this. Now, the gizmo works, if you go to the Data tab, the gizmo works by selecting which one of these you actually want to see in the environment. You can click on all of them or just keep the 4 that they give you. And then you can go ahead and see what happens over a course of years. You have to decide personally how many years you want to look at, right? So let's say I wanted to look at just normal conditions, didn't change anything for these 4 species over 20 years. I would click this button 20 times, and it would show me the conditions over 20 years. And I can see that over 20 years, I'm starting to see the sponges are starting to do better, all right? But most of the organisms are staying about the same in terms of their population size. Okay, but changing any one variable in this, such as, let's say the temperature, if I was to change the temperature up to 32 degrees Celsius and do the same 20 years, wow. So we can see quite a difference there, right? My sponges are kind of the first to go. They're all really going down, my corals, and then my algae is actually doing really well over 20 years. It's the only species that actually benefited from that. Right, so changing these different settings and the amounts of years will change how the population's growth curves do, and we can tell a lot about those populations through that. So let's go ahead and take a look at the first example here for task 1. Okay, so I'm going to go ahead and get rid of this stuff because this is what, when I did this with you guys earlier, this is what we kind of came up with. Now again, the examples that I'm going to come up with here are things that I don't want you to use exactly what I do. Okay, so what I'm going to do is kind of a example. You should use it as sort of a tutorial, but definitely not copy it, because if you do copy it and use the exact same things, even if you go through the motions and do it, I'm not going to count it, because I want you guys to come up with your own sort of version of this and your own approach. Okay, so the first thing that you'll see when you come in is introduction. Yay, welcome to the Food Web Lab. Okay, it's going to tell you what you're going to be doing. And then our first task is, based on the following scientific question, create a hypothesis, variables, control, and constant, collect data using the Coral Reefs Biotic Factors Gizmo, and come to a conclusion. Okay, so we are going to start with our scientific question. What is more impactful, disease or land use on the coral reef populations? Okay, so we're going to go back to our gizmo. So we are asking what is more important, disease or land use to impact these populations. When we talk about impact, the first thing we need to figure out is what do we mean by impact, all right? Are we talking about impacting them in a good way or impacting them in a bad way? Typically, of course, we usually in ecology talk about impacting in a bad way, right? Because we are trying to make sure that the ecosystem doesn't crash. So let's say impacting in a bad way. So what's a good way to measure that impact in a bad way? Well, when we looked at our data before, we could see that a lot of the populations were starting to go down when we changed certain factors. So what if we said that impact was the number of populations that were going down? Right, so that's how we would measure impact, is the number of populations that would decrease over time based on a variable versus those that would increase or stay the same. Okay, the second thing we have to decide in our hypothesis is whether or not we think that disease or land use is going to be more impactful on the populations. Okay, now in the last class that I did, we decided that we thought that disease was going to be more impactful. Again, this is your hypothesis, so it doesn't really matter whether or not you're going to be right or not. There's no right answer. We're just going to go ahead and assume that disease is going to be more impactful. So my hypothesis starting out, right, is going to be that disease is going to be more impactful on the coral reef populations than the land use. Now again, we need to go to more impactful, and remember when we said more impactful, it's not specific enough, because how do we measure more impactful, right? So we have to come up with a way to measure this. Okay, so we're going to say that disease is going to cause more populations to decrease than land use. Okay, and we could say more populations in the coral reef to decrease than land use. Okay, so basically we're saying that the disease is going to tank more populations in this coral reef. Now, how much disease is the next thing we have to determine, okay, and which disease? All right, so specifically I'm going to choose the white band infection rate, okay, so I'm going to go ahead and say the white band infection, okay, disease. So that's the disease, the specific disease I'm going to go for, and I need to figure out what percentage of that disease I'm going to apply. Let's go ahead and say 50%. Okay, so I'm going to say that a 50% white band infection disease is going to cause more populations in the coral reef to decrease than land use. Now I have to go over to land use. And in land use, I have to determine, okay, which one of these am I going to choose? I'll choose sewage and take that up to 50% just to kind of match what I did before. Okay, so in this case, I'm going to say then land use, I'll say then 50% sewage. Okay, the last thing I need to add to this population, because now I've specified my type of disease, how much, right, and my type of land use and how much, and what I think is going to be more impactful. Now I have to say over how much time, right, because remember this is being measured in the amount of years. So I'm going to say over 20 years, let's say, because that's a good way to put it. So my hypothesis is now 50% white band infection disease is going to cause more populations in the coral reef to decrease than 50% sewage over 20 years. That is a very measurable hypothesis. It's specific. It tells me exactly what it is I'm looking for. It tells me exactly what I'm going to be working with. It's really great. So now I go down to the independent variables. Now, in this case, I know I said before we'll need to change one independent variable at a time. And normally that's true. But here, and we are going to do that, right? We're going to do two separate trials. One with the 50% white band infection and one with the 50% sewage. But in this case, we're going to be comparing those two different studies, okay? So for our independent variable, the two things that we're going to change are we're going to say 50% white band infection is the first thing we're going to change, and we're going to say 50% sewage is the other thing we're going to change. Our dependent variable is we're looking for the population decrease, okay? So how many populations are actually going to be decreasing because of our changes to these things. Our control, of course, is going to be the thing that doesn't receive the independent variables. So in this case, it's going to be 0% white band infection and 0% sewage, right? Because I'm not going to be having either of those be there for my control. It'll allow me to compare it to my experimental group. My constants, what am I keeping constant? Well, the cool thing about this is that it actually shows you the things are keeping constant. If I'm changing sewage, then the things I'm keeping constant are logging and agriculture for land use. So I can say 0% for logging and agriculture. Okay, and then the other thing that I can change here is that I can actually go over to my disease, and if I know that I'm changing just the white band infection rate, I know I'm not choosing the black band infection or the sea urchin. So I can say, and then black band and sea urchin. Oops, sorry. Sorry, let me go back. 0% for black band and sea urchin. There we go. Okay. And then, of course, I also want the amount of time, right, to always be the same. And then over 20 years, right? So I need to make sure that I put in that time. Now, I want you to notice something. The constants are everything basically but the independent variable, and it's also going to include the over 20 years. So whenever you're wondering about your constants, just remember it's everything but the variables you're changing, and it should also kind of include the time that you're doing your simulation for. So now I can go ahead and do my test. So the first thing I'm going to test is the white band infection rate. All right, so I want to make sure that my land use is all at 0, okay, because I don't want my land use to be changed at the same time. I don't want to test both sewage and the white band at the same time. I want to see what each of them does separately. So I go to my white band infection rate, put it up to 50, all right, and then just looking at these 4 species, 1, 2, 3, 4, 5, 6, 7, 20. Okay, so over 20 years I can see that all these populations actually increased. The only population that decreased was the staghorn coral. So I have lost 1 out of 4 populations, okay? So I want this graph in order to be part of the data that I use for my conclusion. So I'm going to go up to this little camera right here. I'm going to click on it. It should say processing screenshot. Now you can right click on this, and to right click using your Chromebook, you're going to use 2 fingers and hit the touchpad at the same time. And you can just say copy. And if you want to, you can go straight in here, and somewhere up here, just say Control V, and it'll paste it right in. Then you can resize it so it doesn't take up, the entire thing. You can kind of stick it right here. Now, that was testing my white band infection. Now it's time for me to go ahead and test my sewage. So I'm going to go back over here. I'm going to return to original settings, go over here to land use, sewage up to 50, okay, and restart my years. All right, and now I'm going to go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 20. So here's what this looks like at 20 years. Now we can see 3 populations are going down, and the algae is skyrocketing. So 3 out of 4 populations in this case are going down. I'm going to process the screenshot. Now if it gives you that little message and it won't process the screenshot, what you can do is just press refresh. And it is a little bit annoying because when you do that, it will require you to go back, go to land use, go back to sewage, and then redo everything. But it's not that bad, right? And we get the same results. So we click on the picture and right click, copy. And then over here, paste. We're going to go ahead and make this smaller. And voila. Okay, so we now have our 2 different graphs that we can use for comparison. Okay, now what's our conclusion? So our hypothesis was that white band infection at 50% was going to cause more populations in the coral reef to decrease than 50% sewage. But looking at these graphs, it looks like actually the opposite is true. It looks like our sewage actually made more populations go down than our white band infection. So we're going to go over here to conclusion. We're going to say the hypothesis was not supported. Okay, and that's fine. It's okay if your hypothesis isn't supported, right? You've just learned something new about the world. Whether or not your hypothesis was right, you now know sewage is way more impactful than this particular disease at the same rate. So the hypothesis was not supported, and then we need to say because, right? We need to add in what part of the data actually convinced us, okay? Because only 1 population decreased with 50% white band infection, and 3 of the 4 populations decreased with 50% sewage. Okay, so why do we need to add that to our conclusion? Because that is the data that backs up our assertion, right? We need to show our data. And you might say, well, our graph is already showing our data. No, our graph can't speak for itself. You have to speak for the graph. This graph isn't really saying anything. It's your interpretation of the graph and the context of your hypothesis that makes the big difference. At that point, you're done and you can move on to task 2. Now remember, you cannot copy this exactly what I've done. You need to change certain things. All right, so I don't want to see an exact replica of what I've just done in the assignment. Hopefully, though, this will help you figure out how to do this assignment. Thanks so much, guys, and I hope you guys have an awesome day.